Hello and welcome to Nikolai's genetics lessons, and today I got a question from one of my subscribers. Here's a question: if we cross one genotype on the left with another genotype on the right, what will be genotype and phenotype by crossing at F1 generation with itself? I know the answer even before I finished reading, because this is very easy. We are going to have 10 different genotypes, and we are going to have phenotypes with a ratio 9 to 3 to 3 and to 1. If you want also be able to solve such problems this fast, you need to watch this video till the end. So we have two genotypes. Let me write down once again. The first genotype is capital A, capital C, capital C, capital R, and small p, small p. We cross with another genotype that is capital A, capital C, small r, and capital P, capital P. In the F1 generation, 100% genotype of all the F1 generation. So this is parent 1 here on the left, and this is parent 2 on the right. So F1 generation would be following genotype capital A, because this parent only can give a dominant allele A. For gene A, as you see, this gene represented in diploid organism by two alleles, and second parent also have two dominant alleles. So whether this or this the progeny would inherit, still they would have one dominant allele from this parent and one dominant allele, no matter which one, from the second parent. So we know that progeny for the gene A would have two dominant alleles, and for the gene C, once again, parent 1 has two dominant alleles, parent 2 also has two dominant alleles, so no choice here. For the gene C, also all the progeny would have two dominant alleles. And as for the gene R, as you see, parent 1 has two dominant alleles, so can give to the progeny only dominant allele, but parent 2 has two recessive allele, so can give to progeny only recessive allele. So all the progeny would be heterozygous for gene R, and as for the gene P, parent 1 has two recessive alleles, and parent 2 has two dominant alleles. That means that parent 2 only can give dominant allele P, and parent 1 only can give recessive allele P. So this is going to be genotype of the F1 generation, and now according to our problem we have to cross F1 generation with itself. So we have to cross with another genotype that is the same: capital A, capital C, capital R, small r, and capital P, small p genotype. So what other phenotypes and genotypes we can expect? As I said, this is going to be our answer for number of genotypes, and this is going to be our answer for number of phenotypes. F1 generation would have the same alleles for the gene A, two dominant alleles, the same alleles for the gene C. So that means we wouldn't be able to see any variation in the progeny. For example, if we would take these two genes, these two alleles for the gene A of the parent 1, so capital A, and these two alleles for the parent 2, once again, capital A allele. As you see, all the progeny 100% would have the same genotype, capital A here, capital A here, capital A here, and capital A here. So 100% of their progeny would have the same genotype and phenotype for this gene. So as you see, all the variation that we can get only would be due to this pair of genes of parent 1 and parent 2. And this pair of genes doesn't affect our phenotypes or genotypes in the following generations. So instead of writing these alleles, I would use asterisks in all my following calculations. So basically, for our calculations, we need only this part of the genotype of each parent. So how many different variants of the gametes this parent can produce, and the first variant would be capital R and capital P. So this is going to be the first variant. So capital R and capital P. Second variant would be capital R and small p. So capital R and small p. Third variant would be small r and capital P, small r and capital P, and the last variant would be small r and small p. So this is going to be the last variant, small r and small p. And as you see, the second parent also have the same genotype here. So this parent also would produce the same variants of the gametes. Let me write down once again. So the first variant would be capital R and capital P. Second variant would be capital R and small p. Third variant would be small r and capital P. And the last variant would be small r and small p. So now, when we build a Punnett square, we would see all the possible genotypes and phenotypes that such a cross may produce. So we would have 16 cells, 4 columns and 4 rows. And as I said, we are going to have 10 different genotypes. So we are going to get unique genotypes here that can be seen only once in our table, and the rest genotypes would be repeated at least once or 2 times. For example, all the genotypes that is here in this part of the table, also we can find the same genotypes in this part of the table. So let's count. We have 1, 2, 3, 4 unique genotypes that would be repeated only once, and 1, 2, 3, 4, 5, 6. So 4 plus 6 would be 10 genotypes that also would be repeated, and all these genotypes here would be repetition of what we have in this square, green square. So let's see what genotypes we might have here. And of course, I can put asterisks everywhere. That means that before these 2 alleles, we also may put this allele that is capital A and capital C. So asterisk stand for this genotype. But we are not going to put it everywhere because these two alleles would be the same and doesn't affect phenotype, doesn't affect different variants of the gamete that F1 generation can produce, and we only can see variation in the genes R and P. So we would have capital R here, capital P. We would see capital R here, capital P, small p, capital R and capital R here, capital P and capital P, capital R here, small r, and capital P and small p here. Also we would have in the next column capital R and capital R here, and capital P, small p, capital R here, and small p, capital R and small r here, and capital P, small p, capital R, small r, and small p genotype. So we are half done. So capital R and small r here, and capital P here, and capital R, small r, and capital P, small p, and small r here, capital P, and also small r here, and capital P, small p, the last column, and capital R, small r here, and capital P and small p, capital R, small r here, and small p. I do not like letters P because it's hard to say where P is small and where P is large. So we have small p here, and we also have small p here and small p here. So once again, so genotype here would be small r, capital P, and small p, and small r, small p genotype. So once again, if you would check, you would see that all these genotypes are unique. We only can see them once, but genotypes in green box are repeated. So we have the same genotype here and here. We have the same genotype here and here. We have the same genotype in this box, in this box, and here and here. And as you see, I have found my mistake. Here we have capital R and small r, this is correct, but here we should have capital R and small r. So let me change it. So instead of capital R, we should have small r here. So capital R, small r here, and another one, the same genotype would be this and this, and also this and this. So now we have connected all the genotypes, and as you see, those genotypes that is in green color repeated twice. Those that are in red box repeated once. So 10 unique genotypes: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And let's now find how many phenotypes we have here. So here we have one phenotype. We have capital R, and capital P. This is going to be the same phenotype, 2 dominant alleles and 1 dominant, 1 recessive. In simple Mendelian genetics, this is going to be the same phenotype as in this box. So we can circle it with the same color, and this also going to be the same phenotype, and this is also going to be the same phenotype, and this is also going to be the same phenotype, but this one is not going to be the same phenotype because it's going to be recessive for the allele P. So let's use different color. So it's going to be dominant for the first allelic pair and recessive for the second. And once again, we use red color to circle this phenotype because we have dominant allele R and dominant allele P here, and this is going to be the same phenotype as all this circled with red color. But next would be different phenotype that we circle with blue color. That is going to be dominant for the allele R and recessive for the allele P. And once again, we use red color here. This phenotype is going to be dominant for both genes, and this one too. And now we have a new phenotype that is recessive for the first allele, for the first gene, and dominant for the second. And this one we also circle with the same color. So we circle this one with red color. This phenotype is going to be dominant for the both genes. And now we have a genotype that is dominant for the first gene and recessive for the second. So we circle it with blue color. So let's circle it with blue color. And next genotype is recessive for the first allelic pair and dominant for the second allelic pair. And the last one would be unique. We have here situation when first gene would be recessive and second gene also represented by recessive phenotype and have 2 recessive alleles P. So let's count how many phenotypes we have here. 1, 2, 3, 4, 5, 6, 7, 8, 9. So our ratio is 9 that we circled with red color, to, let's count blue, 1, 2, 3. So ratio would be 9 to 3. Let's now count how many we have circled with yellow color, 1, 2, 3. So ratio would be 9 to 3 to 3. And we also have one unique phenotype that we circled with green color. So as you see, ratio, as I predicted, is 9 to 3 to 3 to 1. Once again, 9 would be dominant for both genes, for gene R and P. 3 we would have that is dominant for the first gene, but recessive for the second. And 3 we have that is recessive for the first gene, but dominant for the second. And 1 we have that is recessive for both genes. And we also can put asterisks everywhere here to designate that all these genotypes would start with capital A and capital C. But because we don't have space, we don't have much time, and this is clear that all these genotypes start with the same genotype, capital A, capital C. So we just omit this beginning of each genotype. But of course, each genotype also have to include this capital A, capital C, because here we have only one capital A and capital C, but each parent should have it here, parent here on the side and second parent on top, so when these gametes would join, we would have two alleles A and two alleles C, which is the same. So I hope my explanation were clear enough. Now you would be able to solve such problems very easily and very fast. Basically, in this problem, what we have done, we started with 4 genes, 4 allelic pairs, but we reduce our problem to just 2 allelic pairs that were heterozygous for each allelic pair. And this is classical dihybrid cross. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.